Hi everyone! For Chapter 13, we will discuss the different types of gene mutations that DNA can have and what are their effects in gene expressions. The chapter will end with a little bit about DNA repair. We won't discuss too much into it, but we'll sort of introduce you into a few things that the cell does to prevent or correct those mutations. But the majority of this chapter will be about the different types of gene mutations there can be. As we know it, any change in DNA sequence that occurs is called a mutation. A mutation can be inherited via mitosis from cell to cell, okay, or via meiosis from one generation to the next, okay. So we're talking about descendants of offspring coming from parents. And mutations in terms of biology can affect us in 3 different ways. Even though we tend to not think of mutations as something that is beneficial, but according to biology, it is. They are something that we must have in order to drive adaptation and evolution. Mutations is what gives organisms that genetic variation that's necessary to, sort of drive that process of natural selection. They have to have genetic variation within a population of species, because if every single organism within a population of species is the same, then when an environmental challenge arrives, let's say a drought or, famine, something like that, all the species will die because none of them will have that one variation that will enable them to be different from others and allow them to survive. That's what natural selection is that you have different slight changes amongst the sort of the organisms within the same population of species, so that you can have someone with something different that can have that, sort of trait to survive. Natural selection occurs because there exists genetic variation, and genetic variation arises from, in a sense, mutations. So without mutations, you're not going to get that variation. So according to evolution and biology sense, mutation is something that's, necessary and essential for species to survive. And of course, even though mutations is something we must rely on for variations, they are also very much detrimental, as we know it, to cells and organisms as a source of many diseases and disorders. We have already mentioned many of those in the first half of the semester of genetics. So other than mutations that occurs naturally, we also talk about mutations that either intentionally or unintentionally introduce into DNA sequences. Research actually highly depends on mutations to study functions and phenotypes of newly discovered genes. And you had a small glimpse of that through your UV radiation experiment with mutant yeast cells. The common approach to this area of research is, if we knock a gene out by making it non-functional with, for example, siRNA or microRNA, as we have already discussed in the last chapter, whatever way we can do some sort of RNA interference to interfere with the gene expression, then we look at what will happen to the cells or organism phenotypically when you knock out that gene. When that protein is no longer there, what happens to the cell? In a way, it's kind of like finding out how a certain part works in a complicated machine. You take away a part from the machine and see whether the machine still runs. If it doesn't, then you further find out why it doesn't run. Is there a particular connection this part you remove serves? So that's what you're trying to find out, okay? In a sense, when we talk about a new gene that we want to find out what it does or serves within a cell or an organism. And that's why science is always involved a lot of problem solving and troubleshooting, in a sense. You're trying to find out and solve a puzzle, in a sense, and figure out what is the best way to discover what is wrong with the cell. It's a very complicated and long-term process, but it is how we now come to find out how all these genes work in our cells. Okay, so there are 2 general categories of mutations. One is called somatic mutation, Somatic mutations means they only exist in somatic tissue, not gametes, okay? This type of mutations cannot be passed on to the next generation of offspring. So it's something that happened in that one organism and it stays there. It doesn't get passed to their children whatsoever. I like to use the example of a mole, okay, like skin mole. Just because we have a particular mole on certain parts of our skin, it doesn't mean our children will have that exact same mole in that very same spot. Some of it could be, environmental driven. Who knows? Okay, so that's why, there are some things that are somatic mutation that is not passed on to the next generation. So somatic mutations are usually passed to daughter cells through mitosis within that same organism. This can lead to very serious consequences if we are talking about somatic mutations that happen early in embryonic development. Okay, so we're talking about embryonic development is when somatic mutations can be very detrimental to the organism. During embryonic development, all of our essential organs are forming their structural framework for their basic functions that we will rely on for the rest of our lives. So that ball of cells after fertilization relies very quick rounds of mitosis to become a multicellular embryo. So the earlier that these somatic mutations occur, the more cells will end up with the same mutation. And it will be even worse if it was to happen to stem cells that have acquired those mutations. So the earlier it is, maybe it was supposed to be heart stem cells, it's supposed to be a liver stem cell within that ball of cells, and all of those stem cells can be mutated if the mutation happened early on before the, the cells start splitting more and more in a process that we call differentiation. But, don't worry about differentiation. But in a sense, that's what we mean by that somatic mutation gets dangerous when the embryo is forming. Okay. Germline mutation. Okay, so that's a second general category of mutations. These are DNA errors that can be passed on to the next generation. That's why it's called germline mutation, because they can pass on to the next generation. They affect gametes, which are your sperm and egg cells. Most of those inheritance pattern of genetics we have discussed in the first half of the semester really is talking about different types of germline mutations. If parents have this genotype and this allele, what is the likelihood that their offspring will produce that same phenotype or have this and that allele? So we did that. We were able to estimate that is because those mutations were germline mutations. If it was somatic mutations, there's no, it's not going to be passed on to the next offspring. We don't have to calculate, okay? So all that we talked about before was some sort of germline mutation. So here's a good diagram that separates the two pretty clearly. So here's your normal adult fish, zebrafish, and then you have somatic mutation that only affects that particular cell within that particular fish. And if that cell undergoes mitosis, because scale, skin, all that stuff is always undergoing cell division, then you may end up with a clump of or population of mutant cells that spreads over the adult zebrafish. But if it's germline mutation, so we're talking about that little egg cell or sperm cell that has that error or mutation in it, and through sexual reproduction, we have talked about how, half of the genes come from mom, half the genes come from dad. So that's why, technically speaking, half of the generation of the offspring fishes will have that same mutation that was carried by this particular egg or sperm cell, and the other half will have no mutation. Okay, of course it all depends on whether that gene is recessive, dominant, etc. But generally speaking, they have half the chance of getting the disease because you have half the genes coming from this particular fish. Okay, so that's why germline mutation in a sense can affect offsprings for generations and generations, because then this fish can technically, if that mutation also affected its egg and sperm cells, then it will will pass on to their next generation as well. Okay. All right, so I recall one of you asked me about this back when we were working on the chromosome lab. So here it is again. This is how we will distinguish between mutations within the chromosome versus a gene. As we have seen from chapter 6, there can be a whole bunch of different types of chromosome abnormalities. Some we can see by eye through karyotyping, as we have done yourself through the lab of karyotype lab, like translocations or deletions or duplications, or even having extra chromosomes or missing a chromosome. That's what we did when we're trying to identify or find a good nomenclature for different types of chromosome mutation. Some we have to rely on computer for more detailed analysis of those banding patterns on those chromosomes, because there's only so much our eyes can do. But when it's like small banding pattern change, we rely on computer. But regardless, those are still considered as large scale genetic alterations, and these are called some chromosome mutation. What we will discuss in this chapter are the smaller scale type of genetic alterations that affect expression of a single gene or a protein, or what we refer to as DNA lesions, because they're technically a break within the DNA, either a break or something wrong with it that it was switched around. Okay, so these are called gene mutations. And just like how we have talked about the different kinds of chromosome mutations organisms can have in chapter 6, remember all those inversions, translocation, tandem duplication. Now we will discuss the different kinds of gene mutation cells and organisms can have. So we will first classify gene mutations by their molecular basis. First type is called base substitutions. And as the name suggests, it is basically that one nucleotide that was switched over to another nucleotide. It is the simplest type of gene mutations because it only involves one single nucleotide difference in the DNA sequence of a gene. There are two types of base mutations. One is called transition base substitution, which is when a purine is substituted by another purine. So from an A to a G, or from a G to a A, vice versa, or a pyrimidine for another pyrimidine. So a T to a C or C to a T. Okay. This is more common type of base mutations or base substitutions because chemically speaking, they are more similar in structure, because purines have 2 carbon rings and pyrimidines has only 1. So it's easy to switch between, chemically similar structures. Now, the other types of base substitution is called transversions. This is when a purine is replaced by a pyrimidine. So from an A to a C or A to a T, or vice versa, G to a C, G to a T, okay? The other way around, C to a C to a G, okay? Or T to a T to a G, okay? So this is when, either a purine is replaced by a pyrimidine or a pyrimidine is replaced by a purine, okay? Switching chemical structure, as you can see, okay? All right. But remember, it is transition that's more common because it's easier to switch between those two. Now, the second molecular type of mutation is called insertion and or deletions. So they are collectively actually called indos because in stands for the insertion, dels stands for the deletion. So when you're either adding one or more nucleotides, insertion, or removing one or more nucleotides, deletion. Okay, this type of mutation is usually more detrimental in consequences than base mutations because it causes what's called frame shift mutations. Okay, from the last chapter, we understood what is considered a codon, and that is made up of 3 nucleotides. When you insert an extra letter into a sequence or delete a letter from a sequence, the reading frame of codons can be completely messed up from the point of insertion or deletion, because those 3-letter codons will have to be regrouped according to their new reading frame due to the extra letter or missing letter. Okay, well, a simpler way to think of it or imagine it in your head is, imagine the sentence. I'm gonna write it. Let me see, where should I write it? I'll write it here and then I'll erase it. The dog ate the yam. Okay, so this is a codon, this is a codon, this is a codon, this is a codon. This is what we mean by reading frame. Okay, where the codons are being segregated or divided is what we mean by reading frame. Okay, and then the yam. So it's supposed to make sense because, these are codons that encode for a specific amino acid that makes a functional protein. But imagine if you were to insert a big letter right here. Let's say an H, and then, insert an extra random letter. So now, all of these codons or reading frames are just going to be regrouped. You have the, and then all of a sudden you have the H inserted into there, okay? Which means the next reading frame will be HDO, okay? And then the dog, the G of the dog, will now be shifted to the next word, ETH, so on and so forth, okay? So now from the point of insertion, everything else after that is reshifted into a different reading frame. So that's why this type of mutation is very detrimental. Okay, the consequence of these frame shift mutation can either make an entirely different protein because everything else after the insertion or deletion will be different type of amino acids, or a lot of times they will actually introduce an early stop codon, which terminates the protein synthesis process early, resulting in a shortened or truncated protein. So let's say it was supposed to be, lots of sequence, and then all of a sudden there's a stop codon, okay, but it's supposed to be a lot more down there. So then your protein process stops here, which means only this part of the mRNA ends up being transcribed, translated. Everything else just stops, okay? They're degraded and deleted, okay? So that's why frame shift mutations is detrimental. Either you're creating something completely different, or you end up with a shortened protein that's no longer functional. Okay, so let me erase all that stuff. Okay, so because codons are in groups of 3 nucleotides, there's some good news to deletions and insertions. If one ends up inserting 3 extra letters or deleting 3 letters from the DNA sequence, so just in terms of 3 nucleotides, then there won't be any frame shift of the reading frame. These are called in-frame insertions or in-frame deletion. Excuse me for my typo there. Okay, you still end up with an extra amino acid or a missing amino acid because you are inserting either 3 letters, which is 3 nucleotide of the codon, or you're deleting a codon. So, but at least after the point of insertion or deletion, everything else stays the same reading frame and will encode for the same sequence of amino acids as the original sequence. So at least you're not making something completely different, or you're truncating or shortening the protein from what it's supposed to be. At least you're just inserting 1 extra amino acid, and hopefully that 1 extra amino acid wouldn't interfere with the protein structure too much. Okay, and this diagram here is a good summary of these 3 types of molecular mutation, gene mutations. Here's your original sequence, wild type. Base mutation means you're just switching out that 1 nucleotide T to a C. Okay, so T to a C is that transition or transversion. is a transition. Okay, so make sure you know how to identify that for the next exam. Okay, if I give you sequences, you're supposed to be able to tell me what kind of mutation it is and what specific, if it's base substitution, what specific type of base substitution it is. Okay, now base insertion is then here you have GTA. Okay, so instead of replacing the T, now you're going to insert an extra T, which then shifts all these codon after that. Okay, or you can delete a T, which takes out that one nucleotide base from the codon, and again everything else reshifts after that. Okay. All right, so a third molecular type of mutation is called expanding nucleotide repeats. So this is a more complex form of molecular mutation. This is where a segment of DNA is repeated in extra copies that will increase the amount of DNA that's available. So one possible source of this mutation is formation of hairpin structures in the DNA molecule, okay, where your nucleotides in the template strand can be replicated twice, which can lead to expansion of your original repeats. Now, a number of human genetic diseases like Huntington's disease we've talked about does contain this type of mutation. Okay, so this diagram here shows you that whole process, how an expanding nucleotide repeat is generated. So for example, here's a DNA molecule with about 8 copies of this CAG repeat. After, when they're getting ready to replicate, remember the 2 strands separate, okay? And then they will undergo replication, just like that. Now, because these are repetitive sequence, okay, they can form these type of secondary structure, which I refer to as hairpin structures, because they are repetitive sequences. And that's why in research, when we're doing PCR, we'll learn about what PCR is about, but they always say it's hard to do PCR on repetitive sequences because they like to tend to form secondary structures within the DNA molecule itself. And now you have just, took this whole bunch of nucleotides out of these newly synthesized strand. So now the DNA polymerase think, oh, wow, there's all this from the template strand that we still have to sort of make, add complementary base pair to. So they finish it off and starts replicating and adding new nucleotides for the rest of the newly synthesized strand. Okay, so after DNA replication, and then you separate the 2 strands again because it undergoes another round of replication. Now you have this whole extra segment of DNA sequence that has been hairpinned out in a sense. But when you separate the 2 strands, they will stretch out again like this. So now you have all these extra sequences, which then will be replicated in the next round of replication, because then DNA polymerase is just going to finish it off and add nucleotides complementary to that strand. So now, instead of having 8 copies of that particular repeat, you have 5 additional copies of the CAG repeat because of this hairpin structure. It's kind of hard to imagine. I will upload a animation video to eCampus that will illustrate this concept. But it is because of this formation of a secondary structure that results in the extra segment of DNA that was not replicated, and when it separates again, it thinks is a sequence that you have to replicate, and that's why you end up with extra copies of that particular segment, and that's why it's called expanding nucleotide repeats. That segment of repeats just keep expanding bigger and bigger. A number of human genetic disease, like I said, including Huntington's disease, does have this type of mutation. Now, how many times a segment of DNA was repeated has to do with both severity of the disease and overall stability of the sequence. The more repeats you have, the more severe form of the disease you will have. And the more repeats you have, it is also more likely that it will expand to even more repeats, increasing the instability, in a sense, of the DNA sequence. So the longer more repeats you have, the more likely it's just going to keep forming these secondary structure, like a hairpin structure or even multiple hairpin structures. And that's why it is like, it makes it even more likely to make it even a bigger sequence. So we don't want these expanding nucleotide repeats very much. All right, so a different type of classification exists for gene mutation. So what we've been talking about, the 3 types: base substitution, insertion deletions, indels, and expanding nucleotide repeats. Those are molecular type of classification. We're talking about structural changes. Now, this type of classification, we're looking at their functions. What will happen to the phenotype or to the final protein product if this mutation occurs? So this type of classification is called functional type of mutation classification. So, for example, there's forward versus reverse mutation. Forward mutation is one where a mutation will alter or changes your wild type phenotype to mutant phenotype. If we think back on our yeast experiment that we did before spring break, the original yeast colonies are supposed to be white in color, but we used a mutant strain that makes the colonies red. Remember that ADE negative you wrote in your lab exercise? So that's an example of forward mutation. You change the wild type phenotype, which was white color, to a mutant phenotype, now making red colonies in your yeast cells. Reverse mutation, is the other way around, or sometimes they call it reversion, is when a mutation changes the mutant allele or the phenotype back into a wild type. Again, this is what we were observing with the yeast experiment. We plated red mutant strains of the yeast, and then we introduced mutations through UV light. And some may require mutations that will acquire mutations that will change the mutant phenotype, red, back to its wild type, which was white in color. This is what you had to explain in your lab exercise activity for one of those questions. One of those questions asks, how are these white colonies forming after it's been UV irradiated? Well, some of you mentioned reversion or suppression. So this is what we mean by reverse mutation or a reversion. We'll get to suppression in a bit, Again, reversion means changing that mutant allele back to a wild type allele, on that same mutation spot, A missense mutation is when you have a base substitution that results in a different amino acid in the protein, While a nonsense mutation is when you change a sense codon. Remember sense codon, what are those? Those are the sense codon are the codons that will actually encode your amino acid. Remember, you have 61 of those. 3 of the 64 codons are nonsense codon, which in a sense is your stop codon. So nonsense mutation is when you're changing any of the 61 codons that encodes for an amino acid and change it to a stop codon. And you already can tell what will be the consequence of that. If a nonsense codon it's inserted, then you end up with a termination spot. So if a nonsense codon is close to beginning of the mRNA sequence, then it is definitely a more detrimental type of mutation because the protein will be truncated and become non-functional a lot of times. So this is a good diagram that gives you, or summarize these different types of mutation. When you don't have mutation, you have your normal DNA sequence that give that particular mRNA sequence and amino acid serine. When you have a missense mutation, you're switching that one nucleotide, which is that base substitution, which resulted in a different codon on the mRNA. Now you're encoding for a leucine, okay? Missense mutation, encoding for a different amino acid. Nonsense mutation is you're doing a base substitution still, okay? But somehow it switches it into a nonsense codon, which is a stop codon. Now you stop making proteins, and the protein stops there, and hence your protein cuts off, and now it's a shortened, truncated version of the protein. Silent mutation is when you're changing a codon to a synonymous codon, which then encode for the same amino acid. So this is not so scary compared to the other types of mutation and that's why we call it silent mutation, because even though here you see that it was switched from AT to GC here, but it still gives you UCG codon, and it so happens that both UCA and UCG encodes for the same amino acid serine. Okay, serine in the wild type sequence, and then also serine in the mutated sequence. So it's called silent mutation because it doesn't give a phenotypic effect. Because it encodes for the same amino acid. Now, a neutral mutation is a missense mutation that do alter the amino acid sequence, but it does not significantly change its function. Some amino acids are chemically similar, okay, and that's why sometimes they may not have an effect even though there was an amino acid change. Now make sure you understand the difference between these two, silent mutation and neutral mutation. Silent mutation encodes for the same amino acid even though there was a base substitution, while neutral mutation does change the amino acid in sequence, but that particular amino acid probably have a chemically similar side chain or something, same charge or something like that, same polarity. Or they have little influence on the overall structure of the protein, that the change causes no effect on the final protein's function, okay? So that's the difference between silent mutation and neutral mutation. Make sure you don't confuse those two. Okay. So before we get to suppressor mutations, let's take a break and we will get to part 2 of this chapter.